Hello, my name is Robert Zeiser. I'm the director of the Division of Tumor Immunology at Freiburg University Medical Center. And today I will report on mesenchymal stroma cells for acute graft-versus-host disease treatment. A large number of clinical studies have investigated MSCs for various sources as a treatment for acute or chronic graft-versus-host disease. The major finding is that MSCs are generally safe and well tolerated, and most clinical studies have generated encouraging efficacy results, but the response rates vary highly between the different trials. The mode of action of MSC is still unclear, but there's evidence that intravascular MSC destruction is followed by monocyte-mediated immune modulation, and this is also consistent with a lack of cell engraftment, meaning that MSCs are often not detectable shortly after transfer. But that is irrelevant because the efficacy is mediated by the monocytes that have been differently polarized then. In vitro studies demonstrated strong immunomodulatory properties of MSCs. This was pioneered by the group of Katarina LeBlanc. The clinical outcome of the first patient treated, reported in 2004, showed that infusion of haploidentical MSCs from the patient's mother improved GI and hepatic manifestation of severe graft-versus-host disease. The clinical benefit of MSC infusion was then further corroborated in a European collaborative non-randomized phase 2 trial, using a shared ex vivo cell expansion protocol. And in this trial, 55 patients with severe steroid-refractory acute GvHD were treated with allogenic HLA-identical, haploidentical, or mismatched MSCs. 30 patients of the 55 patients achieved a complete remission, and 9 patients achieved partial clinical improvement. Since then, many clinical trials on MSCs and GvHD have been performed. Meta-analysis that was recently reported by Chen and colleagues studied 301 MSC-treated patients with steroid-refractory GvHD, and in these 301, in 205 patients, responses occurred. A large multi-center phase 3 trial then conducted in the United States between 2006 and 2009 assessed the use of an industrial MSC product called remestemcel-L, failed to meet its primary endpoint, defined as complete resolution of GvHD symptoms for at least 28 days after beginning of the treatment. Efficacy of remestemcel-L was later evaluated in a multi-center prospective single-arm study in 54 pediatric patients with steroid-refractory acute GvHD after allotransplantation. The main efficacy outcome measured was overall response day at day 28, and the overall response in these patients at day 28 was 70%. Based on these findings, on December 18th, 2024, the FDA approved remestemcel-L, an allogenic bone marrow-derived mesenchymal stroma cell therapy for steroid-refractory acute graft-versus-host disease in pediatric patients 2 months of age and older. This was preceded by an approval in Japan in 2003. The Japanese credit rating company obtained orphan designation and license to manufacture third-party MSCs, a product called JR031, which is very similar to remestemcel-L. And the first multi-center trial in 14 patients showed response rates of 92%. And in 2015, the Japanese pharmaceutical and medical device agencies granted approval for JR031, called Temcel, for the treatment of acute GvHD in both children and adults. A major problem seems to be the variability between the MSC batches. And to prevent this variability that leads to inconsistencies between the different trials, a group from Frankfurt, Fucci and colleagues, developed an MSC bank made from a pool of bone mononuclear cells from 8 healthy donors. The cells were cultured in platelet-lysate supplemental media and frozen aliquots that could then be used for further expansion. The MSC product developed in Frankfurt, was called MSC Frankfurt am Main, so MSCs FFM, and received national market authorization in Germany based on the hospital exemption clause of the European Advanced Therapy Medicine Product Guidelines, ATMP. Treatment of the first 69 patients took place in 14 different transplant centers in 6 different countries. Those patients had grade 3 or 4 GvHD, and here the MSCs were given at a weekly dose for 1 to 4 weeks. By day 28, the response rate was 83%, and at last follow-up, 61% were still in complete remission. After receiving MSC FFM treatment, the estimated overall survival at 6 months was 75%, which is considerable for patients with grade 3 and 4 acute GvHD. Currently, MSCs FMF are tested in a prospective randomized clinical trial, the so-called EDUN trial, for patients with steroid-refractory acute GvHD. This is a randomized trial where the MSCs are compared to best available therapy, which is ruxolitinib. The results will inform us on the value of MSCs compared to the current standard treatment. My conclusion is that overall, multiple independent trials have shown activity of MSCs for acute GvHD, and 2 products have been approved in the US and Japan, respectively. With that, I'm at the end, and I thank you for your attention.